Good evening, everyone. Thank you for joining us on a Friday evening. I know that COVID has taken its toll, and this is the new normal. And I think by the time we address you next, things would have turned normal, and we would be speaking to you face to face rather than using e-media. But this is what it has boiled down to, and it is the new normal. So, my name is Dr. Atul, and I'll be speaking about the whole exome sequencing. So, the way we have uh, broken down this 1 hour session is that I'll be first taking an introductory session, followed by Rebecca, who will be taking us through the uh, uh, laboratory workflow, followed by what our experience in the lab has been. And this will be followed by a few case studies, and we'll be taking the question answers in the end. You can keep on putting your questions and queries in the chat box, which has been labeled, and we'll be taking up these queries at the end of the session. Thank you. So we all know what the central dogma of life is, from DNA to RNA and from RNA to protein. And what COVID has taught us is that from RNA also you can reverse transcribe and go back to DNA and amplify it uh, and that is what the all real-time PCR is all about. But today what we'll be discussing more about is that uh, from DNA to RNA and RNA to protein, and what happens if there are changes which are occurring within the DNA. So why genetic screening is needed? We know that over uh, 5 crore people with single gene disorders are there. Out of this, around and over 10 lakh babies born with genetic disorders each year. And 20 to 30% of all infant deaths are due to the genetic disorders. And 1 in 100 babies born with an inherited disease. So if we can uh, see why this disease is occurring, we can look for possible cures and also better management of these diseases. So the inheritance pattern can be an autosomal dominant, which is seen in cases like myotonic dystrophy or Marfan syndrome. It could be autosomal recessive, as is seen in case of thalassemias and the cystic fibrosis. It could be X-linked recessives, such as X-linked SCID, or it could be X-linked dominant, such as a Rett syndrome, or it could also be that the DNA, the nuclear DNA is not at all involved, and it is the mitochondrial DNA which is causing the disease, such as in cases of the Leigh syndrome. So, if you look at the cytogenetic and the molecular genetic testing portfolios, you could be looking at gross abnormalities, which can be looked at by karyotyping, or you could be looking at uh, subtle deletions and duplications, which can be looked by chromosomal microarray or a technique called MLPA, or you can even be using FISH. Or if in case you want to look at small variations, then Sanger sequencing, fragment analysis, or next-gen sequencing is the method of choice. So what is an exome? So exome refers to the portion of the human genome that contains functionally important sequences of DNA that direct the body to make proteins. So what you have is in the human body is the whole genome. So the whole genome is not actually making the protein. So the protein making part is known as the exomes, and sequence of all the exons of the genome together form the exome. So why is clinical exome sequencing important? So based on the human genome mutation database, that is the ACMG database, and the online Mendelian inheritance in man and other databases, the A clinical exome sequencing panel has been designed to cover genes that are reported in clinical and scientific studies to be associated with human diseases. And this comprises more than 9,000 of odd genes which are clinically relevant. So the whole exome sequencing involves the sequencing of around 180,000 of these exons in the human gene, which represents only about 1 to 2% of the genome. So it's not that we are looking at the entire genome. It is only around 1 to 2% of the genome. But then these are all the things which are making the proteins. And these 180,000 odd exons are arranged in about 22 odd thousand genes. So when do we recommend the whole exome sequencing? It is a molecular confirmation of a clinical diagnosis in a symptomatic patient to assess the at-risk family members. And uh, whole exome sequencing may also be considered as the first-line testing strategy for conditions with a high degree of genetic heterogeneity, in which panel-based testing is either limited or not available. And what do you do if you have a atypical presentation of a genetic disorder? So this can then be ordered as a first line testing uh, for the patient. So sequencing of the exome is thought to be an efficient method to analyze a patient's DNA to discover the genetic cause of the disease and disabilities. And around 85% of all known diseases causing mutations are located on the exons. So you need not be going in for the whole genome sequencing as the first test of choice. So exome sequencing can definitely be looked at as the first method of choice. So if you look at the course of a patient, the patient, if you will have multiple consultations before the actual diagnosis happens, and this is usually done by MRIs, and then a battery of gene test, one followed by the other one, and then maybe the diagnosis is reached or not even reached. So whole exome sequencing as a first line of test has yielded results close to 29.3%, uh, as opposed to a standard care pathway, which has yielded results in only around 7.3% of the cases. So it is also a cost-saving method if it is used as a first-line method of choice. So workflow in the lab involves taking a blood sample, extracting the DNA, sequencing, and then analyzing it. And what we'll be doing today is that we'll be looking at this workflow in depth. And what I'll do is I'll request Rebecca. Rebecca represents a company called Thermo Fisher, and her by education she's a PhD in chemical engineering from University of California, Berkeley, and she's a senior product manager and consumers and applications clinical next gen sequencing division, and she's based out of South San Francisco, California. And over to you, Rebecca.
Hello, good evening. Can you hear me okay?
Yeah,
Hello?
Rebecca.
All
Yeah, Rebecca,
right.
you're audible. Yeah.
Thank you. So, good evening, and thank you so much for the invitation to speak today and for the introduction from Dr. Atul. My name's Rebecca Rutherford, and I'm a senior product manager at Thermo Fisher, and I'm part of our clinical sequencing division. And so today I'm here to talk about our Ion AmpliSeq Exome Ready for a fast and simple exome enrichment workflow. So to begin, I'd like to give a quick overview of Thermo Fisher Scientific and why we're the world leader in serving science. With over 70,000 employees worldwide, we bring thought leadership in all areas of science. And we have over 10,000 customer-facing support roles, so we can be there to assist our customers in all regions. Thermo Fisher is committed to advancing science and staying ahead on innovation, with over 5,000 R&D scientists, engineers, and over $1 billion invested in R&D activities. And of course, we believe this investment has paid off, with Thermo Fisher reaching nearly $25 billion in revenue, which means you can trust Thermo Fisher will continue to be a global leader. So now I'd like to discuss Thermo Fisher's ion torrent targeted next generation sequencing technology. The ion torrent next generation sequencing, or NGS, workflow is designed to be simple, fast, and scalable for your changing research needs. The foundation of the workflow is the ion AmpliSeq technology, which is an amplicon-based enrichment method for NGS. The Ion Chef system provides automated template preparation, and for some of our assays can even automate the library preparation. The Ion Gene Studio S5 series is designed to enable a broad range of targeted NGS applications with speed and scalability. It is able to cover from 2 million to over 130 million sequencing reads per run, so you can scale according to your research projects with only one sequencer. And finally, the workflow is completed with optimized data analysis provided through the Ion Reporter software. So the Ion AmpliSeq technology is globally recognized as the gold standard amplicon-based enrichment method for targeted NGS. This simple, versatile technology is based on highly multiplexed PCR, which allows you to target from tens to thousands of genes across different mutation types, including SNPs, indels, fusions, and copy number variants. One of the advantages of AmpliSeq is that it uses very low input amounts of DNA or RNA, and for some assays down to only 1 nanogram. Of course, AmpliSeq panels can accommodate high-quality samples such as blood, cell culture, or fresh frozen tissue, but it also can handle challenging sample types like FFPE. Ion AmpliSeq technology is flexible to suit a variety of different research needs. So it can be used across many research applications, from cancer research, gene expression, infectious disease, to inherited disease research, which is what we'll be discussing more when we get into the details of the AmpliSeq exome. So why is it so important to study inherited diseases? You know, there are over 7,000 known rare inherited diseases that impact nearly 350 million patients worldwide. And like Dr. Atul mentioned, you know, over 20% of infant mortalities can be linked to an inherited condition, and about 18% of pediatric hospitalizations are caused due to a rare disease, which is why research in this area is so critical. Fortunately, there are over 470 approved therapies for these diseases, but there's still a long way to go to provide more options for these rare disease patients and their families. So whole exome sequencing is an important tool for studying complex inherited disorders. So now I'd like to share some more detail on the Ion AmpliSeq Exome Ready workflow. So just to recap, whole exome sequencing is used when researching complex disorders with multiple potential diagnoses. Of course, we know that the exome contains the protein coding or exonic regions of the human genome, And this is incredibly important because while the exome comprises only 1 to 2% of the human genome, it contains as many as 85% of disease-related mutations. And exome sequencing is often used to research diagnostic odysseys where prior chromosomal arrays, single gene, or multi-gene panels have been unsuccessful at identifying a variant related to the phenotype. Since the goal of whole exome sequencing is to identify genetic variants that alter protein sequences, this is a test that can be able to identify variants responsible for Mendelian and common polygenic diseases at a lower cost than whole genome sequencing, but of course is still a bit more expensive than those smaller panels. So the AmpliSeq exome is a fast and simple exome enrichment workflow. Some of the benefits of using the AmpliSeq exome are the high sensitivity detection of rare de novo SNPs, high efficiency sequencing with greater than 90% on-target bases and greater than 90% of bases covered at 20x. One of the advantages of the ION workflow is the ability to have flexible throughput. So depending on your needs, you can run from 1 up to 48 exomes per week, which reduces the need to batch exome samples. And we provide the whole workflow, including automated variant identification with predefined analysis workflows for single, duo, and trio exome analyses with Ion Reporter. Altogether, this workflow is meant to simplify exome sequencing, even for labs who may be new to next-generation sequencing. So one of the key advantages of an amplicon-based exome workflow is the fast turnaround time and simplicity compared with hybridization capture approaches. The graph on the left-hand side of the screen shows a comparison of the hands-on and total turnaround time for a single exome. The AmpliSeq exome requires less than 50 minutes of hands-on time and can be completed in less than 8 hours. Exome kits from Illumina and Agilent require much longer workflows with more hands-on time. There's a little bit of noise in the background. If somebody could put themselves on mute.
Dr. Atul, can you please put yourself on mute?
I am on mute, not from my side.
Shruti, can you check your audio?
I'm on mute as well.
Okay. So there's a little bit of background noise, but I'll go ahead and continue.
Kanchan, can you help us with this background noise? Okay.
All right. From the graph on the right-hand side of the screen, you can see the AmpliSeq exome is able to maintain excellent coverage compared with Illumina's Nextera Rapid Capture Kit, with superior percentage of bases with 20x coverage and superior coverage uniformity. Oops. Let me. All right, so with the entire ION workflow, you can go from DNA to variants as fast as 36 hours. Sorry, I think somebody's advancing my slides. I'm not really sure what's going on with the software right now.
Kanchan, can you please just check what is happening?
Yeah, I'm just checking the charts. I'm just checking the attendees list. Yeah.
Sorry for this trouble.
It's okay.
Yeah, just a minute. I'll just check.
All right, shall I continue?
Yeah.
All right. So with the entire ion workflow, you can go from DNA to variants as fast as 36 hours with minimal hands-on time. A typical manual library prep takes about 6 to 8 hours with less than 50
I'll
minutes
just
of
check.
hands-on time. The Ion Chef can prepare the template in about 16 hours, which fits nicely into an overnight workflow. It takes about 3 hours to generate raw sequencing data on the Ion Gene Studio S5 system. And then finally, it can take between 9 to 12 hours for variant calling and analysis with the Torrent Suite and Ion Reporter softwares. So of course, this is from DNA to variants. As many of you know, the actual interpretation piece can, of course, add more time to this, depending on how complex the phenotype may be. For many inherited disease researchers, it's important to be able to run a trio analysis with the proband, mother, and father in order to more quickly identify true de novo variants and understand inheritance patterns. The Ion 550 chip supports 130 million reads, which allows for up to 4 exomes per run while maintaining high raw read accuracy. So this allows for a trio analysis in a single run. If you need to run fewer samples, the Ion 540 chip supports up to 80 million reads and is an excellent choice for running 2 exomes. One of the advantages of the Ion workflow is the ability to scale with your research needs and sample volume. For higher throughput needs, you can run up to 12 exomes per day and 48 per week. To do this, you will need 2 Ion Chef systems and 1 Ion Gene Studio S5 system to achieve the maximum workflow efficiency. So I know that this slide is a little bit busy, so I'll run you through a quick example. So if you prepared 16 exome libraries on Monday, you could template 12 samples across 3 550 chips on Tuesday and sequence overnight. The remaining 4 samples would go on a 4th chip that could be templated overnight on Tuesday and then sequenced during the day. And then the process would continue as shown on the slide, enabling you to sequence 12 550 chips a week with 8 or 12 exome sequenced per day. The Ion Reporter software has options for several different exome workflows. So the single exome sample analysis workflow is able to identify what is different in the sample from a reference genome. The paired exome analysis shows how variants differ between 2 samples. The somatic paired exome analysis shows what variants exist in a tumor, but not the germline of the normal sample. And the trio analysis allows comparison of shared variants across known related samples. And this software is available both as a local server or in the cloud. So now I'd like to show a quick case study where the Ion AmpliSeq exome was used to analyze inherited mutations in a developmentally affected individual. PCD is a rare autosomal recessive genetic disorder that causes defects in the cilia lining the respiratory tract and other locations in the body. Early diagnosis is really important to avoid long-term complications, but it can be challenging to diagnose due to the phenotypic heterogeneity of PCD. So, how can newly arising pathogenic mutations be identified? In this paper, the workflow began by starting with discovery-based research using whole exome sequencing, followed by verification using Sanger sequencing. So in this study, the researchers isolated DNA from the affected individual and the parents. They did whole exome sequencing using the AmpliSeq exome kit. And they were able to identify 2 DNAH5 mutations. The mother was heterozygous for 1 defective allele, and the father was heterozygous for a different defective allele in the same gene. And the affected child had 1 of each of the defective alleles inherited from their parents. And then this was also confirmed with ultrastructural analysis of the cilia, which confirmed the defective morphology was due to a malformation of the dying heavy chain. Technically, the affected individual did not have a de novo mutation, but was just unlucky by inherited the defective alleles from both of his parents. And to close out, I'd like to highlight some recent publications that have come out in the past year and a half that have used the ion AmpliSeq exome. So the one at the top is a very interesting use case for the ion AmpliSeq workflow. So researchers at Fudan Children's Hospital utilized the AmpliSeq exome to perform rapid trio analyses for critically ill infants, optimizing their workflow to get results in 24 hours. Neonatal intensive care unit and pediatric intensive care unit applications are a good fit for the speed of the ion workflow. And it's also been used recently in a wide range of other research topics, from epilepsy diagnosis, reproductive health and endometriosis, and hepatology. So with that, I would like to close out. Thank you so much for your attention today, and I believe we'll be having Q&A at the end of the call.
Thank you, Rebecca, for walking us through the entire workflow and giving us a good view of how the things work with the S5 system. So as part of the next session, what I'll be doing is I'll be walking through what our experience has been using this system. So what prompted us to go in for this system was that it had a very good turnaround time, which Rebecca has also been stressing upon. And there is no need to actually batch the samples. Even 2 to 4 samples are good to go. We also get requests for urgent sample processing, and which we are able to do because there is no requirement of a batching of the samples. There is a shorter time for sequencing, and there is an end-to-end workflow which is available. This system has been validated across countries, and there are many users, and there are peer-reviewed publications for this system which are available. So as part of our validation studies, because we wanted to fully validate the system before we actually employed this for clinical testing, we took a sample from what we call as the genome in a bottle. So this sample has been extensively characterized and sequenced throughout the world, and we wanted to check our concordance with what is available in the literature. So variants were compared for the control variant calling file generated for the AmpliSeq exome panel, which is available at this link, and we generated our own sequencing data. And what we found was that overall sensitivity, what we could achieve in the lab was around 95%. And the overall SNP sensitivity was around 96%. The observed indel sensitivity was around 66% for this system, which is good for because indels, when you're looking through NGS, you tend to miss out. But for this system, it was around 66 odd percent. And for that, the method of technology, the technologies of choice are things like MLPA or a chromosomal microarray. As part of our further validation, we took samples which we had previously characterized, and we checked that whether we were able to get the same variant again and again. And say, for example, for this system, this was run in triplicate, and we saw that what was the reproducibility of the system. So what we could see was that if we run the same sample around 99 more than 99% of the variants were again picked up, and the true causative variant was picked up in all the three replicates. So this was done again and again for many samples, uh, including those for indels. So, for example, this was one of the BRCA samples in which we had an indel, which we uh, took and then replicated it across uh, in triplicates, and we found that for indels, we could have a more than 98% concordance between the replicates. Similarly, this was a beta thalassemia sample, again for an indel kind of a situation. Again, 97, more than 97% of concordance between replicates. So the test has accredited by CAP, and we participate in the CAP proficiency testing program, which is available from College of American Pathologists. So what parameters do we look for? We look at the depth, the coverage, and then definitely the databases which we consult. While IAM Reporter is one of the key bioinformatics tool which is used, but there are others which we use in the lab before we give out a report. So the ACMG and the AMP guidelines say that you need to look at the population data, the functional data, and also do computational predictions, and all evidence should be assessed and included in the report. So the databases which we cover are exhaustive, and we cover many databases before we actually give out a report. So we also use the QIAGEN knowledge base. If you look at this small yellow circle over here, that is what my knowledge would be. And then if you look at the variants in ClinVar and the variants in GMD, all these are incorporated in this QIAGEN knowledge base, so that we are able to cover most of the knowledge which is available worldwide. So we use things like the GMD, we use databases like CentoMD before we create a report. All these public databases, as well as the QIAGEN databases, are covered, including things like the SIFT score and the other scores which predict whether this mutation is deleterious or not. And depending upon what we are dealing with, in this case, what we are dealing is the ACMG AMP guidelines for germline testing, and we classify the mutation as pathogenic, likely pathogenic, variant of unknown significance, likely benign or a benign. So this variant of unknown significance is actually very important part of any report, because what we are seeing is that yes, this could be responsible for the condition, the medical condition, what you are seeing, but there is not enough scientific literature around it. So as you expand your databases, the number of VUSs which you give out from a lab actually grow. So VUS doesn't mean that this is not implicated for the condition. It is that just that there is not enough evidence which is currently available, and maybe in the coming years, it might be classified as a likely pathogenic or a pathogenic mutation. So this is the kind of report what we give is that we give you a pedigree chart, and we also give you that, okay, this is the mutation which has been found, and depending upon what the guidelines are, whether it is a pathogenic or a likely pathogenic, and then we also confirm this mutation by Sanger sequencing. Sanger confirmation is actually required only when we see a low depth kind of a scenario, or we are not, if it is in a region where we are not very sure whether it is there or not. So then we definitely go in for Sanger confirmation. So incidental findings is something that definitely is part of the report. If we see, according to the ACMG working group recommendation, that if we do search for specific types of the mutations in specified genes listed in these recommendations, uh, and if we find a case, we definitely report out the incidental findings as part of the report. So, as part of the exome analysis, which has been done in-house, a study was done on 304 odd samples across the various therapy regimes, as well as so these are the cases which we have tested for various conditions, including neurology, muscular dystrophies, ophthalmic, hepatic, and others. And what we find is that our yield is more when we see a metabolic disorder, because metabolic disorders are very well classified in literatures. Uh, the In muscular dystrophy, we have a success rate of more than around 25 odd percent, and these data is in line with the expected yield of around 25 odd percent when you are looking at individual cases. The yield can definitely be increased when you are looking at prior cases, because what you have in prior cases is that you have the data available for the father, mother, and the proband. So this is very important that the more we test, the more family members are tested, the yield is likely to definitely go up. So what we find is that the whole exome sequencing spectrum covers a wide variety of disorders, and it has been finding space in all the various streams of science. So what we are working currently on is a carrier-seq validation. So it's an NGS assay that detects more than 420 genetic disorders, and this enables the analysis of 28,000 non-benign ClinVar variants, which has the SNAs, indels, as well as the copy number variations by NGS, and things which are difficult to detect because of the presence of pseudogenes or paralogs such as SMN1, or within a locus such as HB1 and HBA2 for thalassemia. Currently, what we are using in our lab is an MLPA-based approach, but that is not a very confirmatory kind of a thing, because MLPA does tend to miss out certain of these alpha thalassemia particularly. So we are thinking of graduating to a carrier-seq based assay. So our next speaker is going to be Shruti, and I'll introduce Shruti. So Shruti is a certified genetic counselor by the Board of Genetic Counselors of India, and has been practicing genetic counseling for over 5 years. And she did initial training from the Department of Medical Genetics, AIIMS Delhi, and completed her course in genetic counseling from the Department of Medical Genetics, Manipal Hospital, Bangalore. She has been part of various research projects on pharmacogenomics from CSIR, IGIB, as well as from Indian Institute of Science, Bangalore. She has been associated to initiating and establishing genetic clinics in various hospitals across the country. And her specialty includes analysis of family history and genetic information, and determining laboratory services, appropriate genetic test referrals, and counseling to help in making an informed decision. She is currently the consultant genetic counselor with Dr. Lal Path Labs. Over to you, Shruti.
Thank you, Dr. Atul, for the warm introduction. Greetings to everyone. Now I will be continuing with the last segment of this session and present interesting cases showing basically practicalities of whole exome sequencing as well as a role of genetic counseling in the cases that we see along. So to start with, just a general slide overview, taking example of neurology segment. If you can see on the right side, it mentions the overlapping features that we tend to observe in different clinical categories in neurology. As such, you can see dystonia, development delay, ataxia, epilepsy. So they all tend to have overlapping conditions and symptoms. So whole exome sequencing can play a crucial role instead of single gene or even targeted analysis.
Shruti, can you speak a bit louder, please? That will help.
Sure. Is it fine now?
Better.
Okay, so these whole exome sequencing can play a crucial role, which can be done to confirm the diagnosis or for prognosis, even for management or recurrences chances. So now the question comes, how can we incorporate genetic testing as well as counseling in the healthcare system? So first key important point that has been discussed in the previous slides as well is the identification of the indication, why this testing can be done. So once a clinical suspicious or the diagnosis has been done, it is important to know that do we really want to go for a confirmatory diagnosis or what is the reason? Is it like recurrence risk, knowing the recurrence risk for the family planning, or there are the therapeutic indications? So all these aspects remain important, followed by a pre-test genetic counseling session can be conducted. So what exactly happened during the pre-test session is foremost the clinical history and the family history. The family history is taken in the form of pedigree analysis. Also, the concerns and the psychosocial issues constraining to the patient and the families are taken, and all these aspects are noted. So once a proper consenting has been done, then genetic testing can be taken up. So once the results are out, as discussed previously, it could be positive, it could be negative.
Shruti, sorry to disturb you again. Audience is not able to properly hear. You have to be a bit more louder.
Okay, so is it fine now? Am I audible now?
Slightly better.
Okay. So, yes, so as I was mentioning, so once the results are out, so it could be either positive, negative, or variant of unknown significance. So, depending upon the results, we have to do the further workup. So, if it is positive, is it correlating with the clinical indication for the patient? What was the initial indication for the testing? Is it the recurrence risk or the family planning or therapeutic? All these aspects are taken care. Secondly, if it is negative, so is it a true negative or is it something else or other genetic testing could be referred for? And even if it is VUS, what further workup can be done for the clinical workup, for correlation, or maybe familial testing? All these aspects are to be considered once the results are out. So these interpretations are taken up in the post-test counseling with always the psychosocial issues which come across whenever we are dealing with the patient. So whatever is discussed, it is summarized in the form of a GC summary and shared across with the patient as well as the referring doctor. Further, if there is a reason for educational material or maybe research or any clinical trial that is going on, all these aspects are also taken care of. So let us see all these aspects in the form of the cases. So here is the case number one. If you can see on the right side, it shows the pedigree drawing in the form of family history. So here the squares are the males and the females are represented by circles. And the affected individual is always shaded with the important information as per the guidelines mentioned downside. And the different ways how the lines are being connected shows the relationship between all the individuals. So here the main patient or the individual is a 3-year-old child born to a consanguineous couple means the parents were married in the same family. So this a patient had the speech and language delay. The history of seizures were also mentioned. The MRI was normal, and it was suspected for neuroregression as well as global development delay. So with this information, whole exome sequencing was done. So the report revealed the presence of homozygous mutation in a gene known as GAMT, which stands for guanidinoacetate acetate methyltransferase. So it causes the GAMT deficiency or the cerebral creatine deficiency syndrome type 2. So let us know more about this condition. So it basically primarily affects the brain and the muscles. So individuals show intellectual disability, seizures, autistic features, tremors. It could be even severe if early treatment is not taken care of. So the CCD follows autosomal recessive pattern of inheritance, meaning the parents carry one mutated gene copy each but are unaffected. And when these both mutated copies are transferred together in the next generation, the child becomes affected. Further, on literature review, we got to know that there are suggested targeted therapies associated with this gene mutation, such as oral creatine monohydrate intake. So there was a certain treatment protocol mentioned in the literature. So if you can see, there are certain points for dosage. If that was taken care, so it shows for the symptomatic patient, the intellectual disability and development delay got improved by up to 21%. For seizures, 18% got complete elimination, 49% with decreased frequency. And in case of movement disorders, the improvement was seen in up to 60% individuals. So if we club up all this information, so the outcome remained majorly was the management option. So this specific patient was from Turkey and had gone back to his native place by the time report was released. So all this information was discussed with the referring physician in India, who then connected with the local concerned doctor to provide the forwarded management plan. So it actually aided in making the therapeutic decisions for the child forward. Further, since it is autosomal recessive condition, there is always a 25% chance, hence prenatal testing option is also available for the couple. Further, during the counseling sessions, we got to know that they belong to a very close-knit community and marriages in similar families are quite frequent. So extended family screening can also be discussed to bring about more awareness. So with this, let's take another case. So it is about the adult onset condition. The concerned person is a 28-year-old male presented with myoclonic jerks in upper limbs. Presence of cervical dystonia was also mentioned. And during the pre-test counseling session, he informed that he used to have anxiety attacks as well as depression in adolescence, which he felt was due to his physical concerns of dystonia and the jerks, for which he even took the therapy but then stopped. If you can look at the pedigree, it says that family history of seizure for his father was also there, and further, obsessive compulsive disorder or OCD in maternal grandmother was also noted. So, with all this information, sequencing was being done, and we identified a heterozygous likely pathogenic variant in the SGCE gene. This gene is known to be associated with myoclonus dystonia type 11, and it follows autosomal dominant pattern of inheritance. So let us read further about this condition. So SGC associated myoclonic dystonia is a type of movement disorder, and it is characterized by myoclonic jerks, often in neck, trunk, upper limbs, dystonia. Also, non-motor features such as obsessive compulsive disorders and anxiety disorders are also observed. As mentioned, it follows autosomal dominant pattern of inheritance, means there is a 50% chances that it can be passed from one generation to other. However, the tendency to show variable presentation is always there. So with this, when we reviewed the literature, so we got to know there are certain aspects related to treatment. First was the use of zonisamide, which supports the improvement of myoclonus and dystonia as the targeted therapy. Further, benzodiazepines and antiepileptics used to treat myoclonus were also showed the improvement for such patients, and the response to other antiepileptics is more variable. Further, botulinum toxin can be especially helpful for cervical dystonia, and improvement with L5-hydroxytryptophan and other salts mentioned has also been reported. Further, as a side effect, symptoms tend to improve temporarily with ingestion of alcohol. However, the risk of addiction recommends as given by the guidelines is against its long-term use. So the outcome for this case remained that with this case we were able to first confirm the genetic association. Further, as mentioned previously, the patient felt that depression, OCD were his sufferings or the response to his clinical symptoms. But actually, these were actually part of non-motor features because of this genetic condition only. So all these aspects were discussed in the post-test session. Options of management were also discussed. The inheritance pattern was also commented upon. And further, the impact of alcohol and guideline suggestions were mentioned to the referring doctor. So I hope that with these two cases, we can take like important and vital points as a take-home message. So with these cases, we can state that whole exome sequencing can play an important role in diagnosis and workup in certain cases with which further interventions can be explored. Genetic counseling do play an important role in healthcare team to integrate these genetic testing and provide with genetic expertise as well as counseling. Further, the prenatal testing, risk assessment, family screening options can also play a role in assisting patients to resolve their specific queries surrounding specific genetic conditions. So with this, I end my presentation. Thank you.
Thank you, Shruti, for walking us through two very exciting case studies. And with that, I'll open up the session for questions and answers. So I have a few questions. The first question is by Asif. And Asif is asking us that what are the major advantages of AmpliSeq over the Illumina NovaSeq technology for whole exome sequencing? So Asif, the first and the most uh, advantageous of using AmpliSeq technology, according to me, is the rapid turnaround time, and there is no need to do a batching. So if you are using a technology like a NovaSeq, you will need hundreds of samples before you can actually start the run. And in case somebody asks you for an urgent report, then difficult to give it with an Illumina kind of a technology. With AmpliSeq, because there is no need for batching, you can run up to anywhere between 2 to 4 odd samples. I'm sure this is the technology to look for if you're looking for an urgent report. So Rebecca, if you want to add, please go ahead.
I think you did an excellent job summarizing. You know, AmpliSeq's advantages really are in the fast turnaround time. And I think we just need to be a little clear on, you know, the AmpliSeq is, you know, the library panel technology that's run on the S5 system. So those two together give you the whole fast turnaround workflow. Because AmpliSeq is also available for Illumina, but you don't get the benefits of the fast workflow because, like Dr. Atul said, on those NovaSeq systems, you still have to wait to batch your samples for it to be cost effective.
Thank you, Rebecca. So our next query is from Dipanjana, and Dipanjana has two queries. The first is that the high false positives that we get for indels and small insertions, which right now are part of the CES workflow. Yes, definitely there is a lot of concern around indels and small insertions and deletions when it comes to the CES workflow. But then there are methods in which bioinformatically such false positives are taken care of. And whenever we get a small indel or indel, definitely we like to confirm it with Sanger sequencing. The second question which Dipanjana has is that what about pair end reads when compared? Uh, so the ion AmpliSeq doesn't have a paired end read technology, while the Illumina does a paired end. So how do you compare the two? So maybe Rebecca, if you can uh, compare it.
I'm not sure I fully understand the question.
So this is about paired end reads of Illumina versus uh, the technology which AmpliSeq is using, the Thermo Fisher S5 is using. So I may add that in Illumina, what you are doing is you are sequencing from both the ends, while in S5, what you are doing is that you are sequencing it uh, from one of the ends, but then you are sequencing both the strands. And the guidelines today state that you should have sufficient depth before you actually report it out. So unless and until you have sufficient depth, which comes from both the strands of the DNA, you are not going to anyways report it out. So I think both the technologies are comparable when it comes to detection of SNPs, and that is what we have seen in various peer-reviewed journals. So Ion Torrent uses a non-optical method for base calling. How advantageous is this for homopolymer errors that we see? So homopolymer region is a region which definitely is a concern for NGS, and as bioinformatics has improved, this concern has really been going down. And if we see something which is covered in a homopolymer region and could be pathogenic or likely pathogenic, definitely Sanger is the way out. And the current guidelines say that you need to do a Sanger sequencing if you see something in a homopolymer region. So that is what we are doing currently in the lab. So the next query is from Sheikh, who says that whole genome sequencing, are prenatal related genes covered? Yes, they are definitely covered. Uh, and whole exome sequencing actually means that anything which is part of your exons are covered, and this helps to you to plan out the next pregnancy for sure. So any correlation between SNPs in human genome and microbial pathogenicity? Definitely, yes. A lot of research is currently going on in that direction, that how does my genome uh, affect the pathogenicity or my uh, 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 susceptibility to the various microbes. So IL-28B was one of the genes which was definitely implicated for HCV, and it made a lot of buzz when HCV was at its peak. That if you have a particular type of genotype for IL-28B, then you are the drug is not going to be work very good for you, and that makes you a bad candidate when you have HCV. So such studies are going on, and we'll be seeing more and more human genotypes being more susceptible to a particular microbe. So, is there any gene therapy for autism? See, autism is a multi-gene kind of a disease, and getting to autism, we might in some cases come to know that what SNP is indicated for this. But to my knowledge, gene therapy for autism is, I don't think that it is available. Rebecca or Shruti, would you like to add?
already summarized. So it depends upon the gene of interest we are talking about. So depending upon those aspects only, maybe we can work on. There might be something maybe in clinical trials or at further aspects, but it will all depend upon the gene we are talking about.
Okay. So thank you, everyone. Maybe we can close this session. We are out of time. It's time for us to wrap up. Thank you for your patience and thank you for being with us on a Friday evening. Thank you.
Thank you, Dr. Atul. Thank you, Dr. Rebecca.
Thank you, everyone. Thanks all participants for this wonderful participation.